Hello students, in the previous class we were discussing regarding a concept of anaerobic respiration. In that we had done with the lactic acid fermentation and alcoholic fermentation, and we told that in alcoholic fermentation the production of ethanol plus CO2 is released, and in the lactic acid fermentation a lactic acid is formed, there is no release of CO2. That means decarboxylation is not there in the lactic acid fermentation, but there is a decarboxylation in the alcoholic fermentation. First point. Altogether, how many ATPs were produced in the case of anaerobic respiration? If a question asked, tell me the answer. Answer is 2. Energetically, the fermentation is not much efficient because number of ATP is only 2, and some hazardous chemicals also produced like alcohol or acids. Clear? Now we will be discussing regarding a concept which is going to produce large amount of ATP, and this happens in the presence of oxygen, and that is called as aerobic respiration. And this happens when the pyruvic acid produced during glycolysis, 2 pyruvic acids produced in glycolysis, and NADH plus H plus produced during glycolysis, they both enter the mitochondria, and the events which is occurring in the mitochondria which we are going to discuss now. Shall I begin that, students? Yes, aerobic respiration. So it is also a fate of pyruvic acid. It is also a fate of pyruvic acid where the pyruvic acid from the cytoplasm, will enter where? Enter the mitochondria. And enter the mitochondria, the events further occurring, everything happens in the mitochondria. And what are the events? What are the major events? I will tell you now. The major steps of aerobic respiration. The major steps of aerobic respiration, these are: one is the oxidative decarboxylation. The second event is called as Krebs cycle, which is also called as TCA cycle. Full form I will tell later. TCA cycle, Krebs cycle, TCA cycle. It is also called as citric acid cycle. The third event which is occurring during aerobic respiration is the oxidative phosphorylation. Oxidative phosphorylation, which is occurring in the electron transport system of inner mitochondrial membrane. So you got an idea now that the oxidative phosphorylation is occurring in ETS of inner mitochondrial membrane. Site I am telling regarding Krebs cycle or TCA cycle or citric acid cycle, it is occurring in the matrix of mitochondria. And the oxidative decarboxylation, it is also occurring in the matrix of mitochondria. Matrix of mitochondria. So, let us discuss regarding all these events one by one. Oxidative decarboxylation, Krebs cycle, oxidative phosphorylation. I already mentioned sites: matrix of mitochondria, inner mitochondrial membrane which has electron transport system. Okay students, the first event which we are going to discuss is the oxidative decarboxylation. Let us begin that. Oxidative decarboxylation. What is the chemical with which this start is? It is a pyruvic acid. The pyruvic acid, we can write pyruvic acid as a pyruvate also. Students, now how many pyruvic acids are there? 2 are there. I will be taking for 1. The same is for another one also. A pyruvic acid having 3 carbon undergo oxidative, means it is going to undergo oxidation. When a pyruvic acid undergo oxidation, it will lose the electron and proton. It is going to lose the electron protons, and this electron proton is lost by the pyruvic acid is going to be accepted by whom? By NAD plus. So, there will be formation of NAD. I will write here now. Yes, NAD plus is going to become NADH plus H plus. How many molecules of NADH? 1 molecule for 1 pyruvic acid. Since there are 2 pyruvic acids, 2 molecules. I will write here now. 2 pyruvic acid means 2 NADH plus H plus is formed. Clear? Okay. The word oxidative over here, but decarboxylation has to come. Yes, the decarboxylation means removal of carbon dioxide, 2 carbon dioxide, because each pyruvic acid will lose 1 carbon dioxide. So, 2 carbon dioxide is lost. Oxidative over, decarboxylation also over, but things are not over. If this to occur, there is a requirement of a coenzyme. That coenzyme is coenzyme A. The coenzyme A, remember the coenzyme A, which requires sulfur for its synthesis, okay, and this entire reaction, very big reaction, leads to formation of a chemical, the chemical having 2 carbon, that is called acetyl CoA. Acetyl CoA having how many carbon? 2 carbon. Acetyl CoA having 2 carbon is formed. And how many molecules? 2 molecules of acetyl CoA has been formed. Clear students? Okay. Now acetyl CoA. So who is doing this reaction? This is done by an enzyme which is not just enzyme, it is a complex of enzyme, because that enzyme requires coenzyme A as a cofactor, NAD plus as a cofactor, magnesium as the cofactor. That means it is a metalloenzyme. It has a cofactor which is a coenzyme also. That means that enzyme's name is what? Tell me. Its name is pyruvate. Pyruvate, because there is a pyruvic acid here. Let me write properly. Pyruvate decarboxylase. No, pyruvate dehydrogenase. Pyruvate dehydrogenase. Pyruvate decarboxylate we had studied in the fermentation process, if you remember. It is a pyruvate dehydrogenase, because pyruvic acid is going to lose the hydrogen. So, dehydrogenase. If you remember, in the biomolecules chapter, we have studied our first class of enzyme called oxidoreductases, also called as dehydrogenases. That category, the pyruvate dehydrogenase comes. I will quickly revise once again. This oxidative decarboxylation happening in the mitochondrial matrix of mitochondria, where 2-pyruvic acid will lose the carbon dioxide decarboxylation and will lose the hydrogen electrons that is given to the NAD plus to become NADH plus H plus, remember, and coenzyme A is required, magnesium is required as the cofactors of pyruvate dehydrogenase. The product is called as 2 molecules of acetyl-CoA, and acetyl-CoA has a 2 carbon. The acetyl-CoA, important word, please remember, acetyl-CoA is called as connecting link. Connecting link. It is a connecting link because it is going to link the product of the glycolysis called as pyruvic acid to the next product, next event called as citric acid cycle. So, it is a connecting link of the anaerobic respiration, sorry, not anaerobic respiration, a link of glycolysis to the Krebs cycle. Krebs cycle. It is a connecting link to the glycolysis and Krebs cycle, whose name is acetyl-CoA, having 2 carbon. Please remember the carbon number. I think things are clear for you. Shall I move on, students? Okay. So, I will be raising this aspect. I will be going for next aspect now. Oxidative decarboxylation over. Till now, what has happened, just students? Glucose had become pyruvic acid. During that, 2 ATPs were formed in the cytoplasm. Keep 2 ATPs with you in the cytoplasm, it was produced by the substrate level phosphorylation in the 7th and 10th step. 2 NADH plus H plus was formed in the 6th step of glycolysis. So, we thought 2 plus 3, so 2 into 3, 6 ATP can be formed, plus 2 ATP by the substrate level phosphorylation. 8 ATP can be the promising production of ATP by the glycolysis. In the oxidative decarboxylation step, how many NADH produced? 2. So, 2 into 3 once again what? 6. 6 here, 8 there, 14 ATPs is expectation till now, 14 ATP. What is going to happen later? Let us see, because we want 24 more ATPs to be produced, and that has to occur during the process called as the Krebs cycle only, because altogether 38 ATPs required. Till now 14 ATPs expectation, still 24 ATP, altogether 38 ATPs are going to be the result of the aerobic respiration. Okay. The next event I am discussing, it is a Krebs cycle. Krebs cycle. Here more and more writing is required, writing and practicing requires. So, even I am writing on the board for that reason only. Krebs cycle. Why Krebs cycle? Because scientist. So, scientist who proposed this cyclic event is called as Hans Krebs. Hans Krebs. So, it is a Krebs cycle. Now, why the remaining names I will tell you. It is also called tricarboxylic acid, means TCA, tricarboxylic acid cycle, TCA cycle. Why? Because there is a presence of a chemical called citric acid formation. Citric acid will be formed, which is a first stable product. Citric acid is the first stable product of the Krebs cycle. Citric acid, which has 3 carboxylic acid groups. It has 3 carboxylic acid groups. So, it is called as citric acid cycle is it's also called citric acid cycle or tricarboxylic acid cycle. Why tricarboxylic? Because citric acid, a first stable product of the Krebs cycle, has 3 carboxylic acid groups. So, first reason, second reason, third reason, it is called citric acid cycle, because the first stable product is the citric acid. If you remember, in the photosynthesis also we tell the first stable product of the Calvin cycle is the 3-PGA. So, the Calvin cycle is called a C3 cycle. The first stable product of the C4 pathway, which is a oxaloacetic acid having 4 carbon. So, it is called C4 pathway. The product in the case of the photorespiration is a 2-PPG, 2-phosphoglycolase. So, we call it as the C2 pathway. Photorespiration as the what? C2 pathway. So, here it is called a citric acid cycle because the first stable product is the citric acid, remember. And where does this Krebs cycle takes place? It is occur in the matrix of the mitochondria is the site of the Krebs cycle. Means all the enzymes required for Krebs cycle are present in the matrix of mitochondria, and Krebs cycle is a unique event of the aerobic respiration. Okay, students, the same scientist called Hans Krebs, he contributed with the Hansliet for the urea cycle also, the urea cycle which is taking place in the ureotelic organisms like human being, which requires cytoplasm and mitochondria also. So, that is the urea cycle also was contributed by Krebs, because urea cycle is also called as Krebs-Hansliet cycle or ornithine citrulline cycle, different names for urea cycle if you remember. Students, let us begin now the Krebs cycle. So, you know that the Krebs cycle has to start with the previous connecting link called acetyl-CoA. So, I will write here acetyl-CoA. Tell me acetyl-CoA has how many carbon? Acetyl-CoA has 2 carbon. Yes, 2 carbon molecule acetyl-CoA. Students, I will be writing how many acetyl-CoAs were produced during the oxidative decarboxylation? 2, but I will be writing for 1. Same thing for another one. That means 2 Krebs cycles will run for 1 glucose molecule, that you should remember. Now, the acetyl-CoA is going to combine. It is a cycle, right? So, cycle means a little one blueprint you should draw, because in the same way if you draw it, it becomes beautiful. So, you can erase like this, faint the background if you write. Okay. The acetyl-CoA is going to react with the oxaloacetic acid. So, acetyl-CoA reacts with the oxaloacetic acid. Oxaloacetic acid is a OAA. It was a first stable product of the C4 pathway. They react here, and this process of reaction between oxaloacetic acid with the acetyl-CoA requires water, H2O. It requires water, and this is done by an enzyme called as citrate synthase. Citrate synthase is an enzyme which is doing this reaction. I am not writing all the enzymes, specific required enzymes only I am writing here. Oxaloacetic acid react with the acetyl-CoA. It is a condensation reaction because oxaloacetic acid has 4 carbon, acetyl-CoA has a 2 carbon. The product which is going to form here, which is called a citric acid. Can I write citric acid as a citrate also? Yes, I can write. The product called citric acid which has 2 carbon. 2 carbon in the citric acid, right or wrong? 4 plus 2, how many carbon, tell me? 6 carbon. The first stable product, citric acid, has how many carbon? 6 carbon. 6 carbon chemical has been formed. Clear, students? Oxaloacetic acid, acetyl CoA, product is called as citric acid. Now, next step, the citric acid undergo isomerization to become isocitrate. Isocitrate. It is the isomerization step. If this is the isomerization step, what is the first step? Tell me. The acetyl CoA reacting with the citric acid, acetyl CoA plus oxaloacetic acid to form citric acid. It's a condensation reaction. It's a condensation reaction where the two molecules are joined here, and it's the isomerization reaction. Isomerization reaction. Okay. I can, if you want, I can mention you that citric acid to isocitrate is done by an enzyme called aconitase. Aconitase is an enzyme here. Okay. Now, isocitrate is going to become now, that's a very important one. This process between isocitrate to the next molecule called as alpha ketogluteric acid. The alpha ketogluteric acid, which has a 5 carbon. Then how many carbons in isocitrate? 5. It has a 6 carbon. That means 6 carbon in this molecule. Here is a 5 carbon. That means 1 carbon is lost. That means there is a decarboxylation. Yes, carbon dioxide is lost. That's a decarboxylation. Okay. It is not only decarboxylation, but also it's a dehydrogenation. So there is a the citric isocitrate is going to lose electrons and protons. Whenever electrons and protons are lost, there should be which chemical? Tell me. The chemical is going to be called as NAD plus, which is going to become NADH plus H plus. So this isocitrate to alpha ketogluteric acid, since there is a removal of carbon dioxide, even the formation of NADH, how can I call this step as? Please tell me. How can I call this step as? This step can be called as oxidative decarboxylation only. Yes, it is the event called as oxidative decarboxylation. Oxidative decarboxylation it is. Remember that. Now, alpha ketogluteric acid has how many carbon? 5 carbon. This alpha ketogluteric acid, you should remember, it comes in the chapter mineral nutrition also, in an aspect called as reductive amination, which you will be studying later. Alpha ketogluteric acid will once again undergo same kind of event. And what is that? There is a removal of the carbon dioxide. From the alpha ketogluteric acid, electrons and protons are removed. That means it is a dehydrogenation. It is an oxidation step where that means product, that will be formation of NAD plus will become NADH plus H plus will be formed, and the product will be called as, I am writing here, students, it's a product is called as succinyl CoA. You know acetyl CoA, right? It is a succinyl CoA. That means, do you feel here coenzyme A is required? Yes, the enzyme requires cofactor as coenzyme A. Clear, students? What can we call this step as? It is, since there is a carbon dioxide removed, NADH formed, this is also a oxidative decarboxylation. Oxidative decarboxylation. I can tell it is successive to successive oxidative decarboxylation takes place in the Krebs cycle now. Clear, students? Okay. Succinyl-CoA has been formed. Succinyl-CoA has how many carbon? Tell me now, students. Since there is a carbon dioxide lost, right? The succinyl-CoA has 4 carbon. 4 carbons are there. Now what happens? Let us understand. The succinyl-CoA undergo substrate level phosphorylation, SLP, substrate level phosphorylation. I will use the same color for all the events. It is undergoing SLP, substrate level phosphorylation. It is only one substrate level phosphorylation occurring in glycolysis, during which what happens? Just see here, student, during the conversion of succinyl-CoA to the succinic acid, succinyl-CoA to succinic acid formation. During this process, a molecule of GDP, guanosine diphosphate, is going to become GTP, guanosine triphosphate. Guanosine triphosphate also can yield ATP energy. 1 GTP equals to 1 ATP. How? Just see carefully here. I will write that event here, okay. After formation of GTP, 1 GTP has been formed. This GTP undergo hydrolysis to become GDP plus inorganic phosphate. This inorganic phosphate will react with the ADP to become ATP. That means altogether 1 GTP has formed 1 ATP. So, 1 GTP is equivalent to 1 ATP, please remember. Now, succinic acid has formed. Succinic acid has how many carbon? Because there is no decarboxylation, right? It is having 4 carbon. One in point you should remember, there cannot be a further removal of carbon dioxide, because the oxaloacetic acid has to form later, also has 4 carbon. Yes. Succinic acid. The succinic acid undergo next event, important event it is. The next event, the succinic acid is going to become a product which is called as fumaric acid. Fumaric acid. Succinic acid is going to become fumaric acid. So, what kind of event it is? I will tell you. Wait. Succinic acid to become fumaric acid. The succinic acid becoming fumaric acid, during this process, there is a production of, this is oxidation step it is, where the FAD plus, is going to become FADH2. FAD plus is going to become what? Let me know. FADH2. See, students, here the FAD, you know NAD, nicotinamide adenine dinucleotide. And what is FAD? FAD plus means it is a flavin adenine dinucleotide. Flavin adenine dinucleotide is a FAD, which is derived from vitamin B2 called as riboflavin. So, right here somewhere it is derived from vitamin B2, which is a riboflavin. Okay. Friends, hope you have taken down all these informations, because I am going to erase them, because that should not disturb our discussion regarding glycolysis. So, there is extra information which you should remember. Is FAD plus is the electron carrier? Yes, it is electron carrier. It is a coenzyme derived from the vitamin B2. Yes, I am erasing this, okay. Fumaric acid has how many carbon? Please tell me. Fumaric acid has a 4 carbon. Fumaric acid has a 4 carbon. Succinic acid to fumaric acid, FADH to has been formed. Tell me, what is this event? It's an oxidation step. It's an oxidation step. Here, when I wrote the first step there, I wrote the enzyme name called as citrate synthase. When I wrote the isomerization, I wrote the enzyme aconitase. Here once again I am writing one enzyme name, because these enzymes are important. Only important enzymes I am writing. The name of the enzyme here is succinate. Succinate, because here succinic acid is there. Succinic acid can be written also as succinate. Succinate dehydrogenase. Succinate dehydrogenase is an enzyme. So, the succinic acid is going to become fumaric acid by oxidation step in the presence of succinate dehydrogenase. Remember, succinate dehydrogenase is the only one enzyme of Krebs cycle not present in mitochondrial matrix freely. It is a membrane bound enzyme. Succinic acid dehydrogenase or succinate dehydrogenase is a membrane bound enzyme. Please do not forget that. Fumaric acid is formed. The fumaric acid is going to become now the malic acid. Malic acid. The malic acid has how many carbon? Tell me. 4 carbon. Malic acid 4 carbon. And what kind of reaction? It is a hydration step. Water is added. It is a hydration step, not dehydration. It is a hydration step. Hydration. Hydration step it is, not a dehydration. Remember, fumaric acid becomes malic acid. Malic acid will once again become oxaloacetic acid. By what kind of step? During this process, it's an oxidation step. Okay, it's an oxidation step. It's an oxidation step here. During this oxidation step, it's a production of NAD plus will become NADH plus H plus. So, NAD plus becomes NADH plus H plus. I just write once again here. NADH plus H plus. So, it is a product of the oxidation. Students, hope this all the things are clear for you. It is the Krebs cycle. I will write here, it is the Krebs cycle. I will be explaining it once again now. But before I explain, students, before I explain, somewhere I just want to write a mnemonics for you so that you can remember it very easily. I will write here, okay, mnemonics. I will take this much place for the mnemonics. And what does that? See now, students, it starts like this. O, citric acid is a single word. O, citric acid is a simple, silly, funny molecule. O, citric acid is a simple, silly, funny molecule. How? See here. O, oxaloacetic acid, citric acid, oxaloacetic acid becomes citric acid. It is a connecting link that we should remember. O, citric acid is isocitrate, A, alpha ketogenic A, simple succinyl CoA, silly succinic acid, funny fumaric acid, molecule malic acid. So, O, citric acid is a simple, silly, funny molecule is the best way of remembering the citric acid molecules. To cross check in the exam, let me once again explain this. See students, here the oxaloacetic acid having 4 carbon reacts with the acetyl CoA has 2 carbon. Condensation reaction in the presence of water here to form citric acid. Citric acid has a 6 carbon. Citric acid becomes isocitrate by isomerization. Isocitrate undergo oxidative decarboxylation, removal of carbon dioxide, removal of electron and protons to become alpha ketoglutaric acid having 5 carbon. Alpha ketoglutaric acid becomes succinyl CoA by once again oxidative decarboxylation. Succinyl CoA becomes succinic acid by the substrate level phosphorylation, where there is a production of GTP and GTP, 1 GTP equivalent to 1 ATP. Succinic acid becomes fumaric acid by oxidation step done by a succinate dehydrogenase enzyme, a membrane bound enzyme, inner mitochondrial membrane enzyme, and a product is a FADH2, which is the electron carrier. Fumaric acid becomes malic acid by hydration step. Malic acid once again becomes oxaloacetic acid by, tell me why, oxidation step, where there is a production of NADH plus H plus. All together, how many NADH plus H plus produced in the Krebs cycle? Let us understand. For one acetyl CoA, 1, 2 and 3. Since there are 2 acetyl CoA for 1 glucose, I can tell 6 NADH plus H plus has been formed for 2 pyruvic acid, means 1 glucose molecule. 1 glucose molecule forms 6 NADH plus H plus. And how many FADH2? 2 FADH2 has been formed for 1 glucose molecules. So, 6 NADH plus H plus, 2 FADH2 has been formed. How many ATP formed? 2 GTP formed, that means 2 ATP formed for 1 glucose molecule. This all the things has to be remembered. We will be writing down all those things, aspects will write down, and I will be erasing this aspect called as Krebs cycle now. Hope you have taken down important enzyme like citrate synthase, aconitase, succinate dehydrogenase. If you remember them, it is enough. Done. Students, before I move on, I just want to ask one question to you. For that, you have to give the answer in the web portal. Succinate dehydrogenase or succinic acid dehydrogenase, this enzyme you already know. Where you have studied about that? And this enzyme is inhibited by a inhibitor. I have given the clue also. And what is that inhibitor's name? Can you just tell me? What is the name of the inhibitor which is inhibiting the succinate dehydrogenase? And what kind of inhibition also you can just mention to me? And that means if the succinate dehydrogenase is inhibited, Krebs cycle stops from where? It will not be called as a Krebs cycle because it is going to stop, because there is an inhibitor, it stops it. Please give me a comment on these aspects. I will repeat the question. Which is the inhibitor? And if inhibitor present, Krebs cycle stops where? And which all the molecules are not formed? Please think and tell me. Now, what I am going to write is overall equation, the overall equation in the matrix of mitochondria. I am writing here now overall equation happening in the matrix of mitochondria. That means it includes oxidative decarboxylation, it's including oxidative decarboxylation and even the Krebs cycle. Oxidative decarboxylation and Krebs cycle together I am discussing now. Let us begin that. See students here, pyruvic acid. Pyruvic acid, I am taking it for 1 pyruvic acid, and we can take it for 2 pyruvic acid because 1 glucose forms 2 pyruvic. Let me write for 1 pyruvic acid now. 1 pyruvic acid having 3 carbon, right, plus what? Plus I am writing NADH, NAD plus. How many NAD plus? In the oxidative decarboxylation, if I take it students, here for 1 pyruvic acid 1 NAD plus. If I take 2 pyruvic acid, then it becomes 2 NAD plus. I will write now 2 here, so 2 NADH plus H plus is formed here. So, I will write here 2 that. What about Krebs cycle? In Krebs cycle 3 places NADH formed. That means we can have 6 NADH plus H plus. 1 was formed during conversion of isocitrate to alpha ketogluteric acid. One more place alpha ketogluteric acid to succinyl CoA. The one more was malic acid to the oxaloacetic acid. So, I am writing 8 NAD plus how many FADH2 formed? That is happening only in the Krebs cycle, right. So, in the Krebs cycle there is a formation of 2 molecules of FADH2. So, let me write that also here, 2 molecules of FADH2 has been formed. How many ATPs are formed? But ATP is formed during glycolysis, the Krebs cycle, and 2 GTP means 2 ATP has been formed during the step called as succinyl CoA to succinic acid, substrate level phosphorylation. So, plus of 1 minute students, not FADH2 here, FAD plus 2 ADP I will write. 2 places we have seen addition of H2O. So, I should write for 2 pyruvic acid since it is 4H2O. 4H2 I am writing the overall equation for 2 pyruvic acid. The product let us write down. The product formed here is 1 by 1, 1 below 1 I can write also. It is a complete oxidation, but the fermentation was incomplete oxidation. What is a complete oxidation? All carbons of pyruvic acid is going to be broken. So, there is a production of 3 carbon dioxide. Since there are 2 pyruvic acid, I can write 6 carbon dioxide. 6 carbon dioxide. 6 carbon dioxide plus what? Plus 8 NADH plus H plus 2 FADH2 plus 2 ATP. Clear friends? So, that means what all the products ultimately from the Krebs cycle, tell me, 6 CO2, 8 NADH plus H plus, 2 FADH2, 2 ATP. If I take all this together students somewhere here, only by matrix, in the matrix, excluding the Krebs cycles, excluding the glycolysis, how many ATP we can tell? Let us think about it now. So, I can write here also students 2 NADH plus H plus into 3 if I make it here, it becomes 6 ATP here. Clear, right? 6 NADH plus H plus into 3 if I make it here, 18 ATP here. 1 FADH2 can produce 2 ATP. So, into 2 we should make it here, means 4 ATP here. 18 plus 4, 22 plus 6, 28 we can tell, right? Remember. So, let us come here to do that. 8 NADH plus H plus 2 FA 2 FADH2. Remember, 1 NADH plus H plus can form 3 ATP. That means into 3 if I make it 24, we can write 24. Clear? And into 2 if I make it here 4 over. And we know that in addition to this 2 ATPs are formed, right, by the substrate level phosphorylation. So, plus 2. So, how many over 24 plus 4, 28 plus 2, 30 over. And for your idea, I will writing here, this is occurring in a matrix, means it is occurring only in a mitochondria. Contribution is by mitochondria here, by mitochondria. You know that in the glycolysis event, you have idea that there is, which is occurring in a cytoplasm, where 2 ATPs are there, already. So, I will write here 2 ATPs are there. With this 2 ATPs are formed by substrate level phosphorylation, plus 2 NADH plus H plus is also there, into 3 if I make it. So, 3 into 2, 6 ATP, 6 plus 2, it altogether becomes 8 ATP. 8 ATP plus 30 ATP here. So, expectation is going to be yes 38 ATP. 38 ATP. The 38 ATP can give us 686 kilocalories of energy. 686 kilocalories of energy can be produced with 38 ATP, because calculating like 1 ATP can produce 7.3 kilocalories of energy. Like 1 ATP can produce 7.3 kilocalories of energy. There will be little loss of energy also. This randomly it was taken. Clear, right, students? But one important event I have to mention here, and what is that? Till now with us, how many ATP we have? Means after the end of Krebs cycle, how many ATP we have? If you ask me, I will tell you we have only 4 ATP. Which one? Those ATPs produced regarding substrate level phosphorylation, they are present now. Which is that 2 ATP formed here during glycolysis, 2 ATP formed here during the Krebs cycle, 2 plus 2, 4 ATPs are present with us after the Krebs cycle, because 2 plus 2. Remaining 34 ATPs, huge number of ATPs are yet to form. That event of formation of large amount of 34 ATPs is called as oxidative phosphorylation. Don't be confused with the word oxidative decarboxylation. There is no further decarboxylation steps. The oxidative phosphorylation, a last event of aerobic respiration, can yield us 34 ATP that is happening in the inner mitochondrial membrane. That we have discussed. Clear, students? And who is forming those 34 ATP? Energy rich molecules. Name of those energy rich molecules who are going to form 34 ATP are NADH plus H plus and FADH2. These are capable to form 34 ATP. How? These molecules, how many NADH plus H plus we have, tell me? We have 8 NADH plus H plus in the matrix, 2 in the case of the glycolysis, 8 plus 2, 10 NADH plus H plus we have. How many FADH2? 2. So, 10 NADH plus H plus, 2 FADH2, both undergo next event called as oxidative phosphorylation. During that process, they undergo oxidation, they remove the electron and proton from them, and that ultimately leads to formation of chemiosmotic hypothesis to form 34 ATP. That event we will discuss now. Okay. So, before that, hope this matter is very clear regarding energy budget. Okay. I am going to erase this and entering into oxidative phosphorylation. I just want to show some photographs to you. This is the organelle where aerobic respiration is taking place. He is the Hans Krebs who discovered the Krebs cycle. Here is a Hans Krebs, he himself has written the citric acid cycle. Very clear, you can see here the oxaloacetic acid, citrate, all that. So, this citric acid cycle we had discussed already. Now, we are going to the electron transport system where there is a formation of 34 ATP. So, that event is called as oxidative phosphorylation. I will just give you the introduction for this oxidative phosphorylation now, then remaining aspect we will take it in the next class. First point, it requires electron transport system. Electron transport system. It requires electron transport system ETS present in inner mitochondrial membrane. Inner mitochondrial membrane. Yes. So, it is happening in the electron transport system present in the inner mitochondrial membrane. First important point over. Second point, here 10 NADH plus H plus 2 FADH2, these were formed, undergo oxidation, during which electrons and protons are ejected out, undergo oxidation. Then these electrons are carried, they are carried by electron carriers. The same event is seen in the case of photosynthesis also. Electrons are carried by electron carriers and ultimately given to whom? Given to oxygen. which creates a process called as proton gradient, and which leads to a process called chemiosmosis, and because of the chemiosmosis there is a production of altogether 38 ATP. It is a brief summary of the oxidative phosphorylation I have given, where these molecules are undergoing oxidation. The electron released by them is carried by electron carriers, which is present in the electron transport system, and ultimately the electron is given to the oxygen, and during which the proton gradient is formed, and it leads to chemiosmosis to produce 38 ATPs. And who are all these electron carriers? How many complexes are present? How the electrons are carried from the one electron carrier to another electron carrier? How the proton gradient is established? How the ATPs are formed by chemiosmosis, which we are going to discuss in the next class. Until then, keep studying, students. Let's meet in the next class. Thank you.